I'd like to thank the organizers and SENS Research Foundation for the opportunity to speak to you today about some of our research at the Institute for Regenerative Medicine. What I'm going to do today is talk not so much on aging, but tissue engineering in association with regenerative medicine, which all can be used in terms of trying to tackle a lot of the problems that we're seeing with aging. So today I'm going to talk a little bit about tissue engineering ovarian follicles. And we have approached this in a couple of different ways. I'm going to talk about three specific little studies that we've done with this tissue engineering project. And two of them are associated with hormone production of ovaries, and one is associated with producing mature oocytes from engineered ovary tissue. So one of the problems we're trying to address is the function of the ovaries. And that function can decline in many different ways in terms of aging, surgery, premature ovarian failure, which has several different causes, both medical and pathological. And the result of this decreased function of the ovaries is the deprivation of the sex hormones, in this case estrogen. And there are a number of ways that you can approach this in terms of therapies and the hormone replacement therapies, which come in many different forms. Injections, pills, creams, gels, all being used in terms of this hormone replacement therapies. But there are a number of drawbacks. One of these is that there is no feedback loop with the ovary endocrine axis. That you can see here, very complicated interaction, feedback loops. So with this drug-based hormone replacement therapies, you kind of bypass this, so you get spikes during the treatment. And so this can result in a number of different clinical complications, including cancers, both breast and uterine, cardiovascular problems. So we think a cell and tissue-based hormone replacement therapy is a better option than what we have at the present time. So we've approached this in terms of encapsulation, and this is using Alginate hydrogels. And it's been used in a number of other different studies. And with this, we can make microcapsules to encapsulate the cells, and we can recapitulate the size of the developing follicle within the ovary. And so our approach is to isolate the endocrine cells, expand them in 2D culture. And this, a lot of work has gone into this 2D expansion because one of the cell types there's a lot of problems in terms of trying to grow them and expand them in vitro, and they start changing and modifying their functional aspects. So we've looked at a number of different things in terms of growth factors, cytokines, serum levels to enhance this expansion to get the best function from these cells. So the overall objective of this particular study is to construct an endocrine tissue with ovarian cells using the encapsulation technique specific to Alginate. And we want to be able to look at its viability and functionality long-term in culture. So this is the overall study design, where we'd isolate ovaries from young rats, in this case, isolate the two stromal cell populations within the ovary, granulosa cells and theca cells, expand those, and then encapsulate those, and then look at their viability and function in culture. And this is just showing in how we isolate those cells. We used a density separation technique in terms of isolating the two populations, you can see there. And on flow cytometry, you can see the enrichment of these cell populations. And growing these cells, after isolating, we can show that these theca cells express LH receptors as well as a lyase, or an enzyme. In this case, it's responsible for the synthesis of androgens, this CYP17. It's part of the P450 subfamily. And so over expansion, we still see the functional aspects of these cells maintained. Then the granulosa cells express follicle-stimulating hormone receptor, as well as CYP19, it's aromatase, and it's involved in the conversion of androgens into estrogens. So you can, again, see that the function of these are still maintained as we expand them in culture. So if we take those, encapsulate them individually, and then place them in culture, separated but associated in the same culture, you can see estrogen is high at the beginning and then decreases, progesterone is low and increases. So we're losing function over time. And with this, we're getting potential differentiation, particularly of the granulosa cells, into a luteal differentiation and increasing progesterone levels. So we wanted to recapitulate what the ovary follicle really does during development. And so with this, you can see the structural components, and they're layered and with the developing oocyte. And so we had the theca cells on the outside, granulosa cells on the inside, and we wanted to recapitulate this in terms of how we were going to tissue engineer this particular tissue. So here we encapsulated the granulosa cells within alginate, placed a polyl-ornithine coat around that, and then again put an outer layer of theca cells, all in alginate. Again, it recapitulates what we see in a normal developing follicle within the ovary. And we've labeled these cells. The granulosa cells were labeled green, a membrane staining dye, and the theca cells red or orange. And you can see that they maintain within this construct their separate positions within that microcapsule. Then we looked at several different schemes of this. One, initially where we encapsulated the individual cells and put them in a co-culture. We then mixed the two cell types and then encapsulated them in alginate. And then we did the two-layer alginate construct, and then looked over time at their viability and their function. And you can see here with a live dead stain, the green indicates live cells, red cells, staining cells indicate dead cells. You can see over a period of a month, we still maintain very good viability in this type of tissue construct. And then looking at function, we can look at the hormones produced. And in the two-layered construct, we get very nice production of estradiol as well as progesterone, whereas the others, different types of schemes that we used for encapsulation gave us decreased amounts of sex hormone production. So just a summary of this part shows that we were able to encapsulate these. They retain their endocrine function and show sustained viability in the encapsulation even for long-term in culture. And the multi-layered architecture gave us the best function of this type. Some of the problems associated with alginate, though, is increased calcification, which you can actually see decreases in the ability of the growth of the hormones to get out of the encapsulation. So it's not the perfect system, but it could be a viable system. So because of some of the difficulties working with alginate, we went to another system to look at the probability of using. It's a collagen hydrogel, which you don't have to cross-link with calcium. With the alginate, we're looking at other methods such as strontium to get away from the potential calcification. But in a parallel track, we're actually looking at other hydrogels, and in this case, collagen hydrogels. So we had a very similar type of scheme for this, where we would have individual cells, thecus and granulosa cells, embedded in the collagen hydrogel as individual cells. Then we used a technique of forced aggregation using Agrowell microplates. And we can aggregate these cells into small spheroids or organoids. And here we mixed the cells together and formed aggregates. And the other scheme is we aggregated the granulosa cells first, and then aggregated the theca cells around the aggregated granulosa cells to give a layered construct. And this just shows the microwells where you can see this is where both are present, and we can produce these very nice spheroids. And here we aggregate the granulosa cells first. Day 2 we come in with theca cells. By day 4 you can see again very nice tight compact spheroid of cells. And this shows over a period of almost a month, 28 days, grown in the collagen hydrogel, and they maintain their structural properties. And again, they remain viable throughout this culture period. And again, we saw very nice functional aspects of production of estradiol in the multi-layered type, less production in the non-layered, and very little production with the single-cell co-cultured. Again, some data very similar to what we saw with the alginate. So, in summary of this one is that we demonstrated that the generation of ovarian spheroid constructs within this microbial system is feasible, and they can be used to achieve sustained release of sex hormones within this collagen hydrogel. So we have 2 different techniques we can use to tissue engineer ovarian follicles. The other aspect of this is the production of oocytes. Because in infertility, there are problems with decreased maturation of oocytes, and pregnancy is difficult in these individuals. So we wanted to be able to generate constructs where not only could we produce the sex hormones, but we could also mature these oocytes within our tissue construct for fertility purposes. So this slide just shows some of the problems associated with the assisted reproductive technologies. And here, live births as women age really decreases in reproductive technologies. And some of the risk and complications are listed here in terms of drugs that have to be used, the side effects of these drugs, ovarian hyperstimulation syndrome, and potential multiple pregnancies associated with this. And there's a large cost associated with all of these technologies. So the objective of this is to develop an ovarian cell culture system that could lead to the production of oocytes. So again, we use similar types of technologies that we use for the hormonal replacement therapy studies within this system. So this just shows a study design where we isolated ovaries from very young female rats, digested the tissue, and here we used a 40-micron screen to eliminate any developing oocytes within our cell system. So we didn't want to start with oocytes present in the system. We took the remaining cells that are less than 40 micron in diameter, aggregated them in an Agrowell microculture system for 5 to 7 days, and then put them into a collagen culture for another 5 to 7 days as a maturation step, and then looked at the morphology and characterized what came out of these experiments. So this just shows where we isolate fresh ovarian cells, less than 40 microns, and they did contain cells that were of germline lineage in terms of expression of MVH, which is a germ stem cell marker, as well as SOX2. They began to form these very loose associating colonies with oocytes associated with them, developing oocytes after 5 days. And you can see a scanning EM there of an oocyte associated with this. But these were very fragile types of colonies. So we then used the Aggrawell system in terms of force aggregation, and then looked over a period of time, up to a week. And you can see the stromal cells around the developing oocyte in our cultured system. This is a native follicle, isolated fresh from a rat ovary. So it's very similar types of morphological characterizations. We also showed that these zona pellucida markers, ZP1 through 3, were present within our tissue-engineered constructs. So the oocyte that's in there is starting to mature. So this shows after the Aggrawell and then further maturation within a collagen hydrogel, that you can see a very nice maturing oocyte, even with a polar body after culture in this type of system. We then looked at the maturing eggs or oocytes, and then looked at their ability to be fertilized with sperm. One of the things we saw with this type of engineered construct is that the oocytes that were produced, their zona pellucida wasn't as thick and well-formed as a normal oocyte out of the ovary. And because of that, we saw a lot of polyspermia when we added sperm to that. But we did find about 37% of the oocytes within this tissue construct was able to produce 2 pronuclei with sperm tail, so they were able to be fertilized. So additional studies now that are ongoing in terms of taking these and producing live births are ongoing. So here we've been able to develop a culture system where we know that germ cells exist. We can then use this tissue-engineered technology to mature oocytes, and it may be feasible to use this for fertility instances in the future. So in conclusion, again, just a restatement of that, we have tissue-engineered techniques for cell-based hormone replacement therapy, as well as production of these mature oocytes. So there have been a lot of individuals associated with this project, and we greatly acknowledge the support of Jack and Pamela Egan for the research here, as well as the SENS Research Foundation for the support of a number of summer students that have worked on this project throughout the time. With that, I'd like to thank you.